All right, so we've got receptor proteins. Oh man. Now I got a protein called an enzyme. What's that do? Does it only break down?
No, it speeds up.
Well, it can break down, it can speed up, it can build up. Okay? Let me give you an example. I know y'all thinking, oh my gosh, you ever gonna shut up? Okay, so answer me one question though. Are you learning anything?
Yes.
All right, are you learning anything that's useful?
Yes.
Anything that's bringing back memories of part one?
Yeah.
I feel like I'm going to learn a lot more than I did in part one.
Okay, so plasma membrane. Enzyme. All right. This protein is going to function as an enzyme. As you know, enzymes, their job to help speed up reactions, okay, help build up or break down molecules. So, let's go back. Use my favorite, table sugar. All right? Now, when we eat table sugar, okay, not such my favorite, okay, it's going to be a glucose and a fructose together. They're held together with their bond. Can my body use that one molecule? Can it use the molecule that is connected as a glucose and a fructose together?
No.
No. One thing that has to happen for cells to use a product, they have to be in their simplest form. So this means, and in this case, when we talk about sugars, the only sugar the body can utilize is glucose. So I've got a glucose and a fructose now. So I'm going to have that molecule be able to get that bond broken, and my enzyme is going to help with that. So this protein that's acting like an enzyme, that molecule, if it fits into this protein, it will come in, it'll attach at the active site, and that product, that function of that protein is to break the bond. Do you guys remember dehydration and hydrolysis, the building or breaking of bonds? Which one is occurring here? Hydrolysis or dehydration? Hydrolysis. The body has to provide a water molecule to break this. Okay? So now I have my glucose, which can go and be used for energy somewhere. What happens to my fructose?
Wait, can you explain hydrolysis really quickly?
Okay, when you have enzymatic reactions occurring, and if we look at having, like for example, the glucose and the fructose and the bond, okay? The way the bonds and the molecules are arranged, okay, to break this bond, I'm going to need a water molecule. Now, that means I have 2 hydrogens and 1 oxygen. That protein will take this water, and it will split, because hydrolysis means, lysis means to break, hydro means water, okay? So it's going to break the water molecule. So what it'll do is it will take an OH and an H. Do I not still have H2O here? But it will split this molecule and provide OH to one, H to the other. And I have now split my sugar molecule. So, I've now split my sugar molecule. My glucose gets to go woohoo, 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 to a cell that might need it. What about the fructose? Fructose has got to be rearranged. The body needs to rearrange the structure into glucose and use it. Kind of cool. And it will. It'll do that. So, I've got enzymes. The enzyme could actually break it down, or it could be something it needs to build up. Make sense? So, now, the ion channel. Made me think about the one I watch on TV. No. Okay, so, an ion channel. Like I was saying earlier, outside the cell, we have lots of positives. The inside of the cell is negative. Sodiums, potassiums, to make sure that the cell has electronegativity, this little bit of electrical charge to it, for the most part, okay, for the most part, potassiums have a channel that's called the leak channel. Nothing's blocking it here, nothing's blocking it here, and potassiums will move across, back and forth. There's also the movement of sodium, but sodium's not as great. Okay, it moves potassiums much easier. So we've got leak channels for the ions. This helps to create the negativity, this little bit of electrical difference across the plasma membrane. Now I've got gated channels. What does it mean to have a gate? It has to open and close. So there is a gate. If it has to open and close, I wonder what's going to make it do that? Any ideas?
A key.
Well, if we're thinking about it being ion-based, we're going to have something happen. So, when we look at the gated channels, okay, we mentioned the receptors. I talked about those. The second messenger system, that's the G protein complex. I did the enzymes. Channel proteins. Oh boy. What have I got going on right here? We're going to find that we are going to be very dependent on something called ligand gated and voltage gated. Uh-oh, what does that mean? Okay, so if I have my protein over here, and it's gated, if this one is ligand gated. First of all, what is a ligand?
It's like a key.
You're probably thinking about the key because of what's going to happen, okay? But what's a ligand? When you ligase something, what have you done?
Put it like Nick.
Huh?
Like Nick.
Break down.
Not split, not break down, do what?
Mix.
What, Kelvin?
Open.
You're going to have to make a connection. You've got to put it together to bind, like a glue. What's an example of one? Well, you're probably thinking about that because of the way DNA is going to have to like come back together or generate like the RNAs and stuff like that, because we do have a ligase and a ligand that works on those DNA strands. Okay. When you were in part 1 and you talked about skeletal muscle contraction. Everybody remember that?
Please tell me yes.
Skeletal muscle contraction. What was the product, the neurotransmitter that started the muscle contraction? Skeletal muscle contraction. The acetylcholine, the ACH. ACH would be a ligand. It's a protein. What produced it?
The nerve.
The nerve. Okay? So, if I had my nerve, is everybody with me? Yes, no, maybe?
Yes.
Okay. Let's think of my whole plasma membrane that I'm creating right here. Let's think of this as a skeletal muscle cell. Okay? So, that cell, that fiber, would have a terminal bud attached to it, right? If this were a cell, fiber, cell, because they're the same, okay? There would be one of these little terminal buds meeting it. And the area where the nerve and the muscle met is called the neuromuscular junction. Does everybody remember that? Yes, no, maybe. Yes. Okay, so the product that got released at the end of that little terminal bud is my acetylcholine. So therefore, if this is the ligand, what happens with the gate? It opens. my gate opened. At this point, what rushes in? So, why?
High concentration.
It's in the high concentration outside my cell. Now remember, inside my cell, I'm now negative. But all of a sudden, I get all these pluses, the sodiums, that get to rush in. What does that do to my charge inside the cell?
Make it positive.
Makes it positive. Is everybody with me?
Mhm.